Our first presenter today will be Maggie Weinroth, who is a second-year PhD student in animal sciences. Maggie hopes to continue to improve public health through food safety. The title of her talk is Fingerprinting, the Hunt for Superbugs in Livestock.
The World Health Organization and the CDC agree. Superbugs are a major public health issue. Superbugs, also known as antibiotic-resistant bacterial infections, are an infection that can only be treated with an antibiotic of last resort, or in the worst-case scenario, they can't be treated at all. It's estimated that 2 million Americans each year has one of these infections, and 20,000 die. Now, what does livestock have to do with this? Well, when food-producing animals get sick, we give them antibiotics, just like we give humans antibiotics. And if you're an animal or a human, the same thing happens immediately after you administer antibiotics. And that is, we see a temporary increase in antibiotic-resistant bacteria. Now, this is a concern because the question is, are our production practices in livestock leading to an increase in antibiotic resistance in the meat that we're eating? While this is a concern, there's not a lot of data on the topic. As a result, that's where my study comes in. The objective of my study is to understand what antibiotic-resistant bacteria is present in livestock production and how it's disseminated through the production system. In order to look at this, I followed 3 groups of animals: one group of cattle, one group of chickens, and one group of pigs. I followed them through one production cycle. So I took feces from when they were growing, meat from when they were harvested, and composted feces from when they cleaned out the pens. From there, I looked at the DNA. More specifically, I looked at the bacterial genetics. Now, bacterial genetics are like our genetics. We can see if there's antibiotic resistance genes present. Now, what's really important here is as bacteria evolve, we see that there's sometimes small changes in the genetic code. While that doesn't usually affect the bacteria, it acts as a genetic molecular marker, so we can see where these genes came from. With these tools, we looked at our research question. Now, my results are still preliminary, but we're finding some really exciting things. In the things that we've already analyzed, we found 1,300 genes in the feces and just under 400 genes in the meat. Some of these genes are present across all 3 species, while some are specific to just one. In the future, we plan to build a library of antibiotic resistance genes and their variants, so that we can understand where antibiotic resistance is in our food production and how it's disseminated through. Once we're able to do that, we will be able to develop things to reduce antibiotic resistance in our food. And as a result, we'll be able to decrease superbugs in livestock. Thank you.